actually be a cause of stuck fermentation, because obviously the killer yeast can kill your primary yeast, but they can also be used as starters to reduce spoilage and again kill wild yeasts and other things that might compete. So a killer yeast is formed by first being infected by a particular virus, in this particular case M28 killer virus. So this virus would then infect the yeast cell and insert its genome into the yeast. The genome is actually a plus-stranded, single-stranded RNA. And so what that means is basically this plus-stranded RNA can be directly translated into protein. So this little thing here is the yeast ribosome. And so the yeast will translate this into a pre-protein that you see here. And then the pre-protein can be taken into the endoplasmic reticulum with the help of certain yeast factors like this and this. And so once this pre-protein goes into the endoplasmic reticulum, or ER, the yeast proteins can mature the protein. They can snip it with an enzyme, which is abbreviated by the SP here, and they can add little sugars onto it, which is these little blue lollipops. And then, so by clipping and chemically modifying it, it prepares the toxin for becoming a mature toxin. So additional yeast factors, these guys here, can then fold the protein and form this disulfide linkage here, this SS bond, linking the alpha and beta fragments of this pre-toxin. Once you've got this folded and chemically modified version of the pre-toxin, it can be shunted to the Golgi apparatus, and that's what you see here, where additional yeast proteins will clip it and modify it so that you end up with just the alpha and beta fragments linked together by this disulfide bond. Once it's in this form, this is the active form of the toxin. It can be packaged in a vesicle or a secretory vesicle and then shot out into the world. And then this mature toxin could punch holes into rival yeasts. So this slide is just an overview of some of the issues that we might have with wild yeasts, like Hansennia spora and Clecera, which look like this under the microscope, very similar to Brettanomyces, actually. These guys are cold and sulfite tolerant, and so can certainly cause problems that are difficult to remove. They produce large amounts of ethyl acetate, which is the nail polish remover. Over here you see additional wild yeast called Pichia. Pichia is a lot more round, quite different in appearance than Hansennia spora and Clecera. But one of the interesting things about Pichia is they're obviously associated with rotted fruit, and so obviously you can minimize the likelihood of that being a problem by being careful in what fruits you throw into the bins. And also, but also these guys produce 4EP and 4EG, similar to Brettanomyces, under the appropriate conditions. And so again, 4EP is a low sensory threshold and creates that phenolic Band-Aid type smell, and 4EG is more of a smoky clove. So some of the prevalence, distribution, and types of wine spoilage are outlined in these next few slides. And so first of all, the question is, where do the spoilage microbes come from? And obviously they're coming in on the grapes and the picking bins and grape processing equipment and so forth. So obviously sanitation is a big issue. This is a data set from ETS labs, and they were looking at who's coming in on the grapes, what microbes are coming in on the grapes, and so they used a variety of their molecular tools to give a variety of wineries a sense for what types of microbes were coming in, and just so they could get a baseline. And so you see here acetic acid bacteria had a huge range, from just at the detection level, which is 10, to greater than 1 million cells coming in with a mean of about 9,750. What's interesting to note here is that 70% of the samples had acetic acid bacteria at some level. And so obviously you can use this kind of baseline information to figure out whether or not you might possibly expect a problem. Same thing with Lactobacillus and Pediococcus and Brett Zygosac and Pichia and Hanseníaspora. So all these guys are definitely coming in on the grapes to a varying degree. And as you can see over here in the far right column, the percent positive. So they're basically always there to some degree. Barrels are definitely a source of microbial contamination, and we know that they're difficult to sterilize, for one thing, or sanitize is probably a better word, because contamination can be subsurface. And so you can see this electron micrograph of these, these deep pits and so forth in the wood. They can be millimeters deep, and so microbes can hide in these little pockets and avoid being cleaned out. So again, they're difficult to sanitize, I should say, instead of sterilize. We're not really sterilizing due to limited contact between any sterilizers that you might use and the wood of a barrel. And so again, the typical approach is to rinse with water, often hot water, to get rid of organics, and you could use sort of a strong base like peroxycarb, for example, which is a great way to remove any soaked-in VA. But those things can also be damaging to microbes. And so again, and then you can combine that with citric acids to sort of kill microbes, but also reacidify the barrel from the prior strong base that you use, the peroxycarbonate. So, but again, the idea is to sanitize the barrels, let them soak, but then let them dry pretty well as well. New barrels, depending on the level of toast, provide wood sugar to microbes as a nutrient source, and particularly wood sugars like xylose that Brettanomyces is able to ferment can be a huge problem. Often there's not a ton of sugar in there, and those are unusual sugars. And so a microbe that has the ability to eat wood sugar would have a nice advantage over the rest of the microbes. And so that's one of the reasons why Brett can be a problem in barrels. And you might as well just throw the darn barrel out because it's hard to kill them if you've got Brett in a barrel. So here's some of the spoilage microbes present during a cold soak, like acetic acid bacteria, lactic acid bacteria, and wild yeast. You don't see a huge diversity of chemicals because things are just starting. These spoilage microbes present during primary become a little bit more diverse. And so again, we still have the possibility of acetic acid bacteria because these are aerobic organisms, and so there might be a little bit of oxygen left over that other microbes haven't consumed. The lactic acid bacteria can produce a variety of end products, such as biogenic amines, and usually the biogenic amines will start popping up as other microbes break down proteins and release amino acids into the environment. And so these lactic acid bacteria get a hold of the amino acids and then can produce biogenic amines. And it's possible sulfides as well as the cells start to become starved for nitrogen. If the nitrogen levels aren't kept to a proper level, then obviously the microbes can make sulfide. Hopefully you remember that from the metabolism lecture. And of course, wild yeasts are always a potential problem. And here's some issues that might arise from malolactic. And so again, the lactic acid bacteria, including Coccus, can do some of these things if it's not a well-characterized strain, like producing VA or again, biogenic amines or the THPs, the mousy off flavors, and diacetyl as well, which again is a buttery sort of flavor, which can be desirable, but if the microbes are producing it in excess, you end up with a butter bomb if that's not what you were going for. And then obviously yeasts, some additional yeasts can produce sulfides and aldehydes, which I've talked about, like acetaldehyde, the tart apple, and all that stuff. There's also defects in barrel aging, which I've already talked about previously. And again, I already mentioned Brettanomyces getting lodged in the subsurface of the barrel and producing all its nasty volatile phenols with their super low sensory threshold. And if you have if you have too much headspace in the barrel, acetic acid bacteria can bloom because obviously they're aerobes. And then later on, problems can occur in the bottle. And obviously the bottle's not a very hospitable place. So something messed up, you've got a little bit more residual sugar or something's left over that the microbes can eat. The interesting thing is a microbe like Brett doesn't need residual sugar, doesn't need any of that, can actually live off some of the malic acid, for example, if there's any of that left over. Or even ethanol, which is crazy, right? Brettanomyces can eat ethanol, and so the microbe is one of these guys that can cause a late bloom, a year or more in the bottle, slowly growing, and then you ship out a bunch of cases to your wine club members, and some of them get blooms. It's possible that it's a Brett bloom because of its ability to survive and eat very low nutrients. So some of the case studies I'll jump into.